everybody. Wow. Thank you so much for being here in the closing keynote lecture. So it's a great honor. It's an immense pleasure to be able to present today to Mar Albà, which is one of the role models in computational biology and bioinformatics in Spain and Europe. So to talk a little bit about her, well, she's from Barcelona. She did her PhD in Barcelona. She's a biologist. After that, she moved to London to do a master in bioinformatics, probably one of the first masters that were in bioinformatics, right? And then she also spent some time, I have here my cheat sheet, right? So she spent some time at the University College London as a postdoc working on a little bit of comparative genomics and viruses and developing computational tools for that. Then she came back to Barcelona with Ramon and Cajal Award, and that was the year 2002, and then in 2005 she became a career professor, and since then she has been working here. So she has been there for a very long time, and then she has created a lot of knowledge around in the place where she was. So she's actually, as I mentioned before, a career professor at the Hospital del Mar Medical Research Institute. And well, she also has been working and teaching in bioinformatics at the Pompeu Fabra University. So the interest of Mar is in understanding the birth and death of genes, right? That sounds kind of mystical thing, right? Especially the birth, she said, right? And she has been a pioneer in bringing up some ideas on how this could be happening. We have heard about genome duplication, but she has some other theories of how genes can be created that are more related to activation of particular loci for that transcriptional activity, and then this giving some evolutionary advantage. That was controversial at the time, but she moved forward with these ideas, and actually now she is starting a big project in which she's trying to give more shape to this and combining RNA-seq and RiboSeq data. And I think that this is what she will be talking about today a lot. I was talking for quite a while yesterday with her, and one of the things I learned that I really liked is that she's actually moving around, finding collaborators to do the experiments that she needs, right? Computational biology sometimes is the other way around, so the experimentalists come to you for your analysis, right? But she's doing it in the right way. Okay, so please join me to welcome Mar, and we are really looking forward to your talk. Thanks.
Thank you very much, Anna, for this very kind introduction, and thank you to the organizers for inviting me. I'm going to focus my talk on part of the proteome that has not been studied much yet, which is the small proteome, and which we are starting to see just now. And I'm going to start with this slide. This is from a review from Couso and Patrakim. And here they give an idea, they measure the potential of this small orpheome, these small proteins or micropeptides that may be encoded in the transcriptome as part of long non-coding RNAs or in long non-coding RNAs or upstream open reading frames. So potentially, these would be the numbers of micropeptides we may have being translated, if all these were translated from these molecules, and this is much higher, much larger than the number of annotated proteins. So this is to say that there is a great potential in the transcriptome to generate small proteins that we have not yet detected. Why haven't we detected them? Why are we not yet studying them? This is because the annotation pipelines, typically we have a cutoff of 100 amino acids to annotate the genes. This is because for longer sizes of these open reading frames, they also could occur at random. And I'm showing a plot where they studied which was the longest size of open reading frames in random sequences. This is the average, the mean values, depending on the transcript length. And we see that if we use this cutoff, we are quite safe. We are above this expected randomness, and we can then annotate functional proteins. But of course, there could be many that are below this cutoff and that we are not going to detect. The question is how many, how can we detect them? Are they functional if they are translated? And these are the questions I'm going to address. And obviously, there are small proteins shorter than 100 amino acids that are functional, are important. I'm giving an example here. This protein toddler, which is important for development, is expressed during gastrulation, conserved across vertebrates, and it is only 58 amino acids long. We wouldn't, actually, this was part of a long non-coding RNA. It was not annotated as a protein coding gene initially until the researchers investigated it, and they saw these conservation patterns, and then they did lots of experiments to characterize its functional. It's essential for development. But this is like finding a needle in a haystack to find these small proteins that they may be functional and that we haven't annotated. There are many possible ones. How do we do this? We have to use conservation across species. And then we also need to show that the protein is translated. And of course, this is a lot of work, and this is being done a gene at a time. But luckily now we have techniques that we can use large scale to many sequences. And this is ribosome profiling or RiboSeq, where we can, similar to RNA-seq, to RNA sequencing, but here what we are going to sequence is the RNA fragments that are protected by ribosomes. So where the ribosomes were sitting and where translation was taking place, we are going to sequence those and map them to the genome or the transcriptome, and we'll have clues about the open reading frames, the regions that are being translated. This technique was first developed by Ngoli et al. in the group of Jonathan Weissman at Berkeley, and now it's been used in many different systems, and we are learning a lot about the expression of small proteins, the translation of small proteins, thanks to this. And you can map the reads to the genome, as I was saying, and then you can quantify the level of translation and maybe compare it to the level of transcription, compare it to RNA-seq, for example. But another very important thing you can do with this technique is to discover new translated open reading frames. And this is because in the reads you map to the ORFs, there is a 3-nucleotide periodicity. The reads map in steps of 3 nucleotides, and this is due to the movement of the ribosome across the mRNA in steps of 3, so codon by codon. And this technique has a single nucleotide resolution, so you actually know exactly which codon is being translated once you analyze this data in this way. There's different programs to do that and then extract those significant signatures of translation. And already early works by Ngoli et al. In yeast and then also in mouse identified many of these small translated open reading frames that nobody suspected they were translated, or not to the extent they already saw in those early works. These would be the reads mapped to the mRNAs, to the coding sequence, or in this example, this is a UORF, upstream open reading frame, where there's also many reads of this technique, ribosome profiling, or even in transcripts that only have small open reading frames, have nothing else. You can see signals in different open reading frames, again, indicating that there's small proteins, generally smaller than 100 amino acids that are being translated. And they, in the papers, they concluded that 1/4 of the 5' UTRs show this kind of signature. So of course that's huge, that's massive. It's a lot of putative micropeptides. And most long non-coding RNAs showed some signatures of translation as well, which is really a very strong statement. At the time, still it is a very strong statement. And I'm going to talk about the research that has been done since then in these different types of small open reading frames, of 4 different types: internal alternative frames, This is when they are inside the coding sequence, but in a different frame that is being translated and gives rise to a completely new protein. Alternative isoforms, like intron retention isoforms, we have been studying, and which also can give rise to alternative frames where part of the intron is translated because it's retained and it's part of an mRNA. Upstream open reading frames, these are really very, very abundant. We still know very little about these ones. And also the whole wall of what is annotated as long non-coding RNAs. These are transcripts that contain, in many cases, small open reading frames. Some of these long non-coding RNAs, of course, perform very important functions at the non-coding level, but many others are poorly conserved, have these small open reading frames, and as we'll see in many cases, indeed, there is translation of these small open reading frames. So I'm going to focus on these questions. How abundant are they? Something about the functions, although we don't know so much. And quite a lot about the evolutionary implications, which is an area in which we are actively working. So internal alternative frames, this is quite intriguing, that you can have an alternative frame inside the protein because, of course, how are these two things then translated? How do they evolve? There may be restrictions for the evolution of the second protein due to the evolution of the first protein. It's quite intriguing, but as we'll see, we are detecting these events with the analysis of ribosome profiling data. And actually, this type of events were already noted many years ago by this guy, Susumu Ono. He's very best known for his studies on gene duplication. He wrote this famous book, Evolution by Gene Duplication, where some of you may probably know it. He was proposing different models for the evolution of duplicated genes that would be identical and then would undergo a number of modifications, neofunctionalization, etc. But then later in his life, he looked at another kind of new genes, and this is not so well known. And these are genes that originated by these alternative reading frames. And he was saying this in the abstract of one of these papers from '84. The mechanism of gene duplication is self-limiting because, of course, the proteins are variations, are similar to each other. So how can you then generate all the diversity we see of proteins in life? How can you, just by gene duplication, then all the proteins should be very similar? What happened then at the beginning of life? How all this diversity was generated? So he was interested in these kind of questions, and he realized that by gene duplication, you couldn't explain it all, because then, as I said, all the proteins would be very much more similar to each other. And then, of course, there's other mechanisms, like gene fusion, and other mechanisms by which you can get new genes. But again, in all cases, the genes come from other genes, so they'll be still very similar. And he became very excited about observations about using alternative frames to generate new proteins with completely new sequences. For the protein, would be a completely new sequence. So he proposed, as a source of a truly unique protein, an unused open reading frame of an existing coding sequence. And he was indeed observing some cases in bacteria where, of course, all the information is very packed. So it may be that you have different proteins encoded by the same sequence in the same RNA. And at the time, they called this overprinting, where you build a new protein by using an alternative frame. And the two proteins may exist at the same time, or at some point, maybe you only have the second protein, and then really you have a completely new protein with a new sequence. And this had a follow-up, this other paper by Keys and Gibbs, '92. I like it very much because they were already thinking about these problems of how can new genes originate, and saying, okay, there was perhaps a big bang, like almost everyone is thinking, like at the beginning of life, then the majority of the proteins falls, they emerge then. Or perhaps there's been continuous creation more than what we think. And this mechanism of overprinting, which they also observe in viruses, was a possibility to create completely new proteins. So it was very, very exciting. And they could see that if they compare then the species, closely related to species of bacteria or these viruses, they could see that these events often were species-specific. So they could be these events that created new proteins and that we could see as very recent events. Okay, going now back to the ribosome profiling and all the data we are now gathering with these technologies. We have learned recently in recent papers that have used these technologies in cell lines that actually there are hundreds of cases that correspond to this same scenario in human cell lines. So we probably have many more cases than what we knew about, because as I said, with RiboSeq, we can know exactly about the frame, which codons are being translated. Here, what we see in the region is a signal by both corresponding to two frames, something we then don't see here. And we also see the ATG, the stop codon. So everything is consistent with the translation of two different proteins. No idea what these proteins are doing. They have not been studied yet, but of course, there's a lot of material here to do further studies. Okay, with these techniques, we are also learning about translation of alternative isoforms that may generate small proteins that maybe we had disregarded, thinking that perhaps because there was an early stop codon, this was not going to be translated. But as you see, in some cases, we see evidence of translation as well. So there may be new protein generated by, for example, intron retention. And we have been studying this in fission yeast. First, recovering all the possible isoforms in this species. There were, when we started this a couple of years ago, there were only like 2 or 3 alternative isoforms in the annotations, despite the fact that about 43% of the genes have introns. But little was known about this, the possibility to generate alternative isoforms. So we use nanopore direct RNA sequencing to obtain long reads and then be able to see these isoforms if they existed and quantify them. This is very nice because each read you obtain corresponds to a single molecule in the sample. So it's very, you can quantify the abundance very well. And then we did an assembly. Some of these reads were not complete. We assembled them and identify different types of isoforms. From intron retention to intron inclusion, that's the new isoform will have a new intron, alternative splicing site, differences in the splicing site in the alternative isoform, which is shown in blue, or exon skipping. These were the 4 main classes. And it was amazing because we could see hundreds of cases. Out of the 2 that had been annotated, we could see many more with this technology, with nanopore direct RNA. And 92 of them had what it seemed to us to be a high frequency. They were of high frequency, more than 20% of the transcripts for that gene. So this is very, very unlikely to be errors. This is more likely to have some kind of function. So suddenly we had 92 isoforms that were likely to be functional. Here, by the number of counts, there was some overlap between the alternative and the reference. Of course, in general, the alternative were expressed at lower levels. And then we asked the question, well, these alternative isoforms now we are seeing here in this system, if they translated proteins, and then we'll go and measure it, but if they translated proteins, how would these proteins be? And in general, they would be small proteins. This is the difference between the isoform and the reference, so we are in negative numbers here. In general, they would be small proteins. And we studied specific cases with ribosome profiling data that was available for intron retention. We checked if there were RiboSeq reads and if there was periodicity down to the next stop codon. Like, for example, in this form here, this intron has been retained. We see this isoform using nanopore. And then if we continue here with the reading frame, we hit and stop. And then with the ribosome profiling data, we can see evidence of translation. And this would be a very small protein that apparently is being translated from this isoform this also high abundance isoform in this case for this ribosomal protein. And we had up to 18 different cases with some evidence of translation. So going to different kind of, moving forward to a new, different kind of small open reading frame, the upstream open reading frames, very mysterious, very, very abundant. We still have to understand what the translation in these upstream open reading frames means, but it's very common. So in this recopilation of cases, I'll talk about this at the very end, but this is a very recent paper where results from different studies using ribosome profiling have been put together. And we have here about 3,000 cases that have this periodicity and are very clear cases of UORFs with translation activity. So this is a very big, the UORFs are a very big group. And then also there's the long non-coding RNAs, which are also quite a large group. So what is this translation in the upstream open reading frames? We don't know exactly, but if these peptides then are stable, this would result in many peptides of sizes between 25, 30 amino acids, larger than what you typically obtain with translations of open reading frames in long non-coding RNAs. And one possibility that has been observed in several genes is that these upstream open reading frames inhibit the translation in certain conditions of the downstream of the main coding sequence. This is, for example, from a review, two different mechanisms by which this can happen. Maybe after translating this upstream open reading frame here, the two subunits dissociate, and then you don't have efficient translation of the coding sequence, or there's stalling of the ribosomes here, and the same thing happens. One well-known case is GCN4, which is a master regulator of stress in yeast. And there's two upstream, this has been very well studied, and it's a very well-documented case, where these UORFs are regulatory, indeed. During normal conditions, the UORFs that are most proximal, there are four. The ones that are most proximal to the coding sequence, they are translated at high levels. Here I'm representing the ribosecretes. But if there is a stress, oxidative stress, starvation, different kinds of stresses, then the dynamics of translation here change. This is not translated at efficient levels, the UORFs, and then this translation can take place. And we can see this in coverage of reads, of looking at different experiments. Here in normal and stress conditions, we can see the changes in the number of ribosecretes, in the translational efficiency of this transcript. But is this the case in general for the UORFs, where we see translation activity? Is this what they are doing? We don't think so, and data from other groups also show that this is unlikely to be the case in general, because when we compare different conditions like normal growth conditions and stress, we see that there is actually a positive correlation between what you observe at the UORF and what you observe in the main coding sequence. So if one increases, the other one also increases, according to an increase in the levels of the mRNA. So there's at least in these stress conditions in general, we don't see this kind of phenomena where the UORFs are probably regulatory, and when they increase, the CDS decreases. So what are they doing? Are they encoding small proteins that maybe are functional? Clearly, it seems that in most cases they would allow for reinitiation, or there will be a leaky scanning of these UORFs so that they don't interfere so much with the translation of the main CDS. Do they encode functional micropeptides? We don't really know for most cases, but some studies, some experiments have started to be made in the Weissman group. They did the ribosome profiling in cell lines, and then CRISPR to see the effects of this when you mutate, when you delete the ORFs that are being translated, these non-canonical ORFs. And for the UORFs, in about 200 cases, they had phenotypes associated with the deletion of the UORF. And in 10 cases, they studied where they could interact with other proteins. And indeed, in 5 out of 10 cases, they form complexes with the main protein. So the two proteins interacted. And this may be in a scenario that is more common than we usually think. In other cases, they had independent functions. They were located in different parts of the cell. The two peptides, they did not interact with each other. These would be cases, and probably there are many more, where we have bicistronic transcripts encoding for more than one protein in humans. Okay, I'm moving to the long non-coding RNAs, which is going to be the largest part of the talk, because I'm also going to then go quite a lot into the evolution, the evolutionary implications of these activities, how we think they are related to the birth of new genes. So long non-coding RNAs, although they are called like this, is a bunch of different things. It's very heterogeneous. Some are very, very well expressed, functional, really non-coding RNAs that regulate gene expression or have other functions in the cell. Many others are expressed at not so high levels, tend to be lineage or species specific, and many have these small open reading frames, ATG to stop, that could potentially be translated, and according already to the early studies of Ingolia, they are actually translated in many cases. And sometimes more than one protein could be produced. So going back to the initial studies of Ingolia, he was arguing that they saw a lot of activity with ribosome profiling techniques in these ORFs in the long non-coding RNAs. And they measure different things, but one thing they measure was the translational efficiency of different kinds of transcripts, protein coding, 3' UTR. Protein coding is like the positive control. You should see, of course, signal here. 3' UTR, you should see little signal. And then where are the long non-coding RNAs in this picture? The majority had translational efficiencies that were corresponding to this distribution of the coding genes. So when these ORFs are translated, they are translated at very reasonable levels, which can be assimilated to what you see for protein coding genes. So this could be just protein coding genes that encode small proteins. This is what it looks like. Others would be completely different, and even if they have open reading frames, they are not translated, or we see very, very little signal. So here probably we have different classes of transcripts. We look at data from different, after these Ingolia papers, and this was very exciting because we were interested in how new genes can emerge other than by gene duplication. So thinking about all these long non-coding RNAs, why are so species specific? Could it be that some of them made proteins, and could this be new proteins? And when these techniques appeared, we were very interested in analyzing the data ourselves, and we did it in 2014 for different species for which data had become available. Normally, this data was being used in the papers more to measure like levels of translation compared to transcription, translational regulation, not so much to inspect the long non-coding RNAs, but that's what we were interested in doing. And what we saw is that indeed, according to what Ingolia was reporting, we could see that a high percentage of the long non-coding RNAs had signatures with the riboseq redistribution that were compatible with translation. So indeed, we could have new proteins coming out of this material, new small proteins. The translational efficiency, which we also measure in these different systems now, often was comparable between long non-coding RNAs and coding RNAs. So this seems okay for producing proteins. And what are these ORFs in the long non-coding RNAs? Well, there's a part, I said, one part would be non-coding, of course, but the part that is translated, I think we also have to think of in two different scenarios. One is those that translate small proteins that are conserved across different species. And here we have examples from a review we did a couple of years ago. Before I talk about this example, toddler, that is involved in development, conserved between human and mouse. And these other proteins that you see, they are small, and they are involved in a number of different functions to do, sometimes with the mitochondria, with muscle activity, oncogenes, development. So a number of functions that have been described. And this typically have been observed from transcripts that were initially annotated as long non-coding RNAs. And they had these small orbs with translation activity. And because they also had conservation, they went and studied them, and indeed they found very important, interesting functions for these proteins. But the majority of the long non-coding RNAs are not so well conserved, not even between human and mouse. The majority of them are not. And here I'm presenting data from a paper from the Kasman group, where they use high coverage RNA-seq data to assemble the transcriptomes of different species and then compare them, and in particular understand for the long non-coding RNAs which were the levels of conservation, how many long non-coding RNAs were in different species. And what they could see for the human long non-coding RNAs, this would be the data here of the level of conservation. So to summarize this data, what they could see is that the majority of human long non-coding RNAs would be conserved only at the level of primates, but there would be no conservation down to mouse or even less to other vertebrates. This is not the norm. These are rare cases where you find this conservation down to vertebrates. So we have lots of long non-coding RNAs that are poorly conserved. And actually, by studies we have done and other groups have done, how we understand this material is that many of these species or lineage-specific small orbs that we know sometimes are translated, they would have an encoding origin. So they would come from the genome by a process called de novo gene birth or de novo emergence of proteins. In general, they would be small open reading frames because randomly in the genome, that's what you have. You have a small open reading frame. So no wonder these new proteins are small. And these are, they have two properties. One is that you see that these long non-coding RNAs or proteins, they are restricted to a species or a set of closely related species. You don't find homologs in any other species in the tree. But now, because we have so many genomes, we can be more sure about this, and we can look at the synthetic blocks in the genome where you have conservation of gene order. You can align these sequences if you have closely related species. I can inspect whether we have the ORF, where we have expression of something in this region where we have the putative new gene, and see that in different parts of the tree. And in many cases, hundreds of cases in each species, we see no evidence of activity. So there's probably many new genes that we have not really recorded as such, and many are being still part of this long non-coding RNA group of molecules. Okay, more about this process of de novo gene birth. So from the genome acceptation of sequences that start to be transcribed, we know transcription is pervasive in genomes. At low coverage, we have reads everywhere. Most of the genome is probably transcribed at some level. So it seems pretty easy to have new transcript units. And then many of these will have orbs just by chance. And it also seems easier than we thought that these start to be translated at some level, at least. So this would generate many small proteins, in general small proteins that not necessarily functional at first, that may acquire functions, and then become what we know as we call de novo gene, which when functional, this may be associated with high expression levels, gain of more expression. We should start observing signatures of natural selection. This will tend to persist more over time. So this is similar, if you remember what I was telling you about before, the overprinting and these ideas of the Big Bang or continuous creation of genes, where they also speculated that would have new proteins appearing during evolution by using alternative frames. Here would have new proteins appearing during evolution because of pervasive transcription and translation events that would generate all this material, and then some of these may end up being functional and persisting. Right, so from this scenario where we think, well, all the proteins have other ancestors that are also proteins, at least partially, and domains that come from other domains, folds that come from folds that were there a long time ago. And there's been tinkering with existing genes, but not really creation of completely new proteins. Now we are thinking more in this direction, at least some groups working on this, where probably there's been the birth of new genes throughout evolution. And the genes that we observe today is only, do not represent all the genes at the beginning of life, but there's been new genes appearing through the activities of the genome, as I was saying, transcription, use of alternative frames, different processes. And some time ago, we decided we saw these activities, this model, these ideas were starting to take shape in our minds and in the research of other groups, and we decided to do a very systematic study in mouse using this riboseq data, very high coverage riboseq data we had by 2018 from different tissues and cell lines. And again, re-annotate the long non-coding RNAs now with a lot of precision using the 3-nucleotide periodicity, and see how many had translated small ORFs. Of course, you also need that they are translated at certain levels to see this periodicity and to be sure this would make a protein. And we had more than 1,000 with these properties, with this very clear periodicity. Some annotated, some novel. This would be very similar to the annotated one, so we ended up putting them together. And in general, the ORFs would be smaller than 100 amino acids. So we had this material, and then we said, okay, let's classify them into conserved and non-conserved. This was to gain more insights into this possible process of de novo gene birth associated with this material. So conserve, if we find homologs in any other species, and here we use a lot of species, and non-conserve otherwise, if these appear to be mouse-specific, we don't find the homologs of these long non-coding RNAs translating these ORFs in other species, and also by selection signatures that indicate something, say something about the possible function of these possible micropeptides, using non-synonymous to synonymous SNPs, and then again classifying the groups of proteins into putative proteins into selection and no selection. And we saw these different classes. We saw different classes that probably tell us about this process of de novo gene birth and allow us to do models of how this may be happening. One class, we saw conservation, and we also saw quite a lot of selection in this class. So this probably enriched in small conserved proteins, many of which are not known yet. Nobody's studying them, but they are here, hidden in this group of long non-coding RNAs. Others that were not conserved, quite a lot. A class that was also under selection, under some selection, at least significant selection, and probably this includes de novo species-specific genes, so these genes will already have function, some of them. That's why we see the signatures. But then a large class, and this was very interesting to us, that we are interested in evolution, right? Many people would dismiss this is noise, but a class that was not conserved and neutrally evolving, no signatures of selection. But of course, this class could be putative precursors of new genes, because you also need these precursors. At some point, they need to be created to then perhaps acquire functions at some point. So we could see this class for the first time in this paper, and this generated some interest. And we also then investigated for this class that are probably not even associated with functions, but they just start to be translated during evolution, which we see these first events. If we take those for which we see translation and those for which we do not see translation, this would be orbs in transcripts for which we cannot measure the signatures, but so some of them are not translated. What makes the difference between the two groups? It would seem interesting. What drives these initial steps in the translation of the proteins? And we see that something that is associated with this is the codon usage bias, the composition of the sequence of the orbs. If it's more like what you see in coding sequences, this is more likely to be translated at higher levels. So this is more likely to become a protein. So in a way, this is telling us that the new proteins will be more alike already existing proteins in the codon usage, in the frequencies of the codons, of course, because some tRNAs are more abundant than others, and this is probably is what favors their translation. This more optimal codon usage bias. So this is what the model we have with which we are working. We and other groups characterizing this new mechanism for the birth of new genes, of new proteins, de novo gene birth, where we have background expression. This could seem noise, but it also is material that then can be recycled, can be used for evolution to generate new functional genes that are then subject to natural selection and continue to evolve. And more recently, we have also been studying doing very similar studies in yeast. Yeast is a very completely different system from mouse, of course. Here we have a much more compact genome. We have less genes. They are, in general, much better characterized. And there are examples of de novo genes that, for example, this one here called BSC4. It's a DNA repair protein, and studies using synteny in these genomes, which are very closely related, and you can align these genomes. You see no evidence of this open reading frame. You see no expression here. So this is probably a de novo evolved gene that was already characterized some years ago by another group. So we were interested in quantifying and seeing how many more like this we could measure using RiboSeq, RNA-seq, using these genomic comparisons. And first, we needed to do de novo transcript assembly. We needed to assemble the transcriptomes and identify genes that are not annotated, or transcripts that are not annotated, because the majority, in the annotated fraction, 99% are coding, but we know we could have some non-coding with the small orbs. That is also part of the story. So that's why we needed to reconstruct them first, and we got several hundred for each, even for Cerevisiae, which is very well characterized. We had about 600 in this class that we assembled with the RNA-seq reads, deep coverage. And then, because we had ribosome profiling data, about 1/3 of them actually contain orbs that had translation signatures, and the proteins would be, of course, smaller than for the annotated ones. So these are the novel ones here. They would encode the small proteins. And sometimes more than one protein, as I've shown before. It happens also in mouse. Sometimes there's more than one open reading frame with signatures of translation. And then we use the genomic information, the genome synteny blocks, to check for these conservation patterns and to define a set of de novo genes, probably de novo genes, that could be conserved in only one species, or perhaps even in 2 or 3, but when not conserved, we could see no hints of expression of anything in different conditions in the other species. So we obtained this class of de novo genes according towards the definition of these genes right now. And no wonder the majority of these de novo genes corresponded to these novel transcripts now here, talking about those that translate proteins. So indeed, there's many transcripts that we had not noticed, even in Saccharomyces cerevisiae. Many of them are antisense to other genes. They have orbs. We see translation signatures. And there is an enrichment, a very clear enrichment in very recent events that could have been generated de novo by acceptation of the genome, by transcription and translation events. So I'm going to end with 3 slides explaining a little bit what we are doing now, where we are going to. One initiative in which we have in taking part is the annotation of these orbs, for which we see translation activity. in GeneCode and in other databases. This is a whole effort to analyze data sets that have been generated, put it together, and finally enter these annotations in the databases so that all of you can see them and study these proteins. And this has been led by EBI and by other researchers in the States and in Europe. And we have now this list of about 700. We call them riboseq orbs right now. So these would be orbs where we clearly see these patterns of translation by the periodicity. It's not just that we have rich mapping there, but we see these periodicity patterns that are compatible with translation. And this is for humans, and now this will need to be done by for other species. We have also been quite interested in understanding the implications of these events of translation of the long non-coding RNAs to generate new antigens in cancer. So here I'm plotting, I'm showing results of a given study where they investigated the expression of transcripts coming from non-coding parts of the genome. So this would be long non-coding RNAs. And those that were tumor-specific, and they saw that they could, because some of them can be translated, often they would be presented as new antigens. And if these are tumor-specific, they're not in normal body tissues, then this means that the immune system, if everything is working fine, the immune system will recognize these new antigens. And this is a way to then destroy these cancer cells. And this could have implications also to understand the response to immunotherapy and, in general, the interplay between the immune system and the cancer cells. The cancer cells we have also detected many long non-coding RNAs that are tumor-specific, and then some of these could then produce these peptides that are novel to the body, to the organism, and which can be recognized by the immune system as if they were like viral proteins. We have received funding from ERC recently to study de novo genes, but now focusing more on populations. I've been showing you comparisons of species. I said they are species-specific, they are lineage-specific. But since there are so many, and what's happening in populations, which are the dynamics of gain and death of these genes within the populations? Can we detect SNPs, mutations that are associated with this birth of new genes, perhaps by generating new promoters, new binding sites for the polymerase, new open reading frames? So we investigate this in different systems. This I'm showing here populations of Saccharomyces cerevisiae from different parts of the world that we plan to investigate. Okay, and many thanks. I want to thank I have presented here work from Jorge Ruiz Orera, former student in the lab, studying mainly the mammalian systems, de novo gene birth and translation of small orbs there. William Blevin, Simone Moro, Marta Huertas, and Jose Carlos Montañez, who have been working in the yeast systems and have also presented work from them. Wonderful collaborators at Universitat Pompeu Fabra and Politecnica that help us generate the data, and I really am really thankful that they got involved in some of these projects and help us with them. And finally, thank you very much for your attention and for being with us until here. Thank you.
Thank you, Mar. I think I will start with the questions of the online audience this time. So there is a question about the translation of small orbs in mouse. Again, is there available evidence from mass spec or the peptide protein level? This would be more convincing also with respect with possible functions. That's one question.
Yes, there is. I haven't talked about this because for time limitations to be getting into all the proteomics data. But of course, we and others, we are checking constantly the proteomics evidence for these peptides, and there is a fraction for which there is this evidence. This may vary from say 5 to 20% depending on the experiments we are doing, and this is more or less what we expect for small proteins in general. So if we look at annotated proteins, that have even well-known functions. Often, more often than not, we do not see them in the proteomics data. The longer the protein, the easier it is to have peptides that would be suitable to be detected by proteomics. So it's really, so we see some, so yeah, we can validate the process. We don't know how many more are missed because the proteins are small.
Okay. Well, I think the other question is related to that, so I will move to a question in the audience. Gonzalo.
Hi, very nice talk. I was wondering, there is this tool from the Baker's group using AlphaFold in which they start from random sequences and they hallucinate and then generate foldable sequences, foldable proteins from random things, right? And this is more or less one of the mechanisms I think evolution might be doing, according to what I understood from your talk. They show that it's easier to generate things with alpha helical elements. Have you checked a little bit of the structures, maybe predictions from these de novo proteins, and is it something coherent with what they have found? Is easier to generate de novo, or do you see something different?
No, absolutely. It seems I'm not an expert in this, so that's also something I haven't talked about. But there's been groups doing predictions and doing even some experiments, and it seems easy to generate like alpha helices, and in many cases this is what is predicted, or other small secondary motifs. Yeah, they are predicted to be able to get some structural properties.
Okay, thanks.
Yeah, thank you for the very intriguing talk. Two questions, if I may. One of them, if you translate them into proteins, their amino acid composition look like normal composition, the ratio between hydrophobic and hydrophilic and all of this stuff. This is the first question. The second question, did you or other groups have looked into similar ideas in prokaryotic organisms?
Yeah. The first question, yes, they have biases, and it's very curious. It's universal, the biases they have, like many other things. We have seen these biases in yeast, in Drosophila, in mammals, and in composition. And what it happens is that they tend to be depleted in acidic amino acids, those that are very recent, those that are, let's say, species-specific. And we see translation. But they have differences in their composition, so they tend to be positively charged. Now it's not clear, we still don't know what the implications of these are, but yeah, there are some differences. And these differences are predicted from ORFs in the genome or transcriptomes or random ORFs. So if this is their origin, that's probably why they still have these properties. I have to say that over time, probably these properties change. We do not observe it for all the proteins that are also likely to have a de novo origin, but for which we cannot be so sure. So there could be, there are probably biases. And in prokaryotes, I think because it's more difficult to compare the species, this is not, I think there's a few studies, but we haven't got into that. And also thinking that it would be a little bit complicated to, there's more horizontal gene transfer, for example, and more events that make it a little bit more difficult to study phylogenies.
Thanks a lot. So first of all, hello. First of all, thanks for this great talk. And also want to thank the person online that already anticipated my first question about mass spectrometry and presence of peptides. My second question is about, in terms of studying the functionality of these macropeptides in the future, thinking about applying, for example, mass spectrometry type of techniques. I think one of the potential hurdles that you might identify is the fact that being micropeptides, essentially, proteomics relies essentially on presence of lysine and arginine and tryptic type of digestion or lysine C. So it might be the case that these micropeptides don't have presence of that, so therefore it will be very difficult to see them with traditional mass spectrometry techniques. So I was wondering if you, what strategy you have in place with your collaborator in order to be able to identify and study these very interesting micropeptides in the future. Do you have something in mind or any comment about that?
Yeah, no, we were thinking of doing like more targeted proteomics kind of experiments. I have to say I'm not an expert. I don't know if this will be possible or not. You can select for small proteins from the beginning, but of course, if you don't have the correct amino acids, you will not get the peptides. So really, the CRISPR studies would be a way to see phenotypes, and indeed they have been working, and we see phenotype for these small ORFs. So I suppose, for example, you could do that kind of experiment in cell lines.
Okay, thanks a lot for your answer.
Thank you very much. I was just wondering if you, in terms of interpreting function for your small peptides, have you screened them for known short linear motifs?
Yeah, we haven't, but that's a very good idea, and that's something we definitely can do. Yeah, thank you.
Thanks.
Great talk. Mar, is there any evidence that these small peptides can get modified post-translational?
Again, I don't know. Have no clue.
Okay.
We have another question, online question. Are the novel short isoforms also present in the data of previous studies where, for example, perhaps they have been filtered out assembled transcripts shorter than 100 nucleotides or something like that? Have you checked that?
Yeah, we found these examples in POMBE, but definitely in other studies, depending on how you analyze the riboseq data, you'll find these alternative isoforms and you find signatures there. I guess for intron retention, normally one thinks that they are not going to be translated, they'll be retained in the nucleus, and in many cases probably they are, but you know, in others, it's worth checking this data because you could have surprises and you could see translation of some of them.
Okay, thanks. We have one last question over here.
Thanks. Thank you for your talk. Just wondering how can we differentiate small ORFs with internal non-coding RNAs, because riboseq won't be our friend here, because the ribosome will pass in this area anyways and will make footprint in the reads. If there is a kind of a test for that?
Internal ORFs, you mean like when there's internal, an alternative frame that is being translated, like that case? Yeah. Or you mean?
No, there are like sponge RNAs.
Oh, great.
They are non-coding. They do regulatory role instead of being a protein.
Yeah, sure. I wouldn't know how to find the regulatory role of these sponge RNAs because it's not my expertise, but then I think it's worth checking. Now that we are building these lists, I mean, we are doing it so that it facilitates research for everybody, and you know for sure that your long non-coding RNA you are studying is not translated, or may not. It's not one of these cases where you could have a small protein that has gone undetected by annotation pipelines. You may want to know that. So we are building these now catalogs that will be available from GeneCode, from Uniprot, where you at least can see if there is significant signatures of translation within ORFs in your RNA. Because the majority of RNAs, long non-coding RNAs, long RNAs that you are studying, even short ones, they have ORFs. So potentially, we now know that a large fraction is translated, so it's worth checking that first, yeah.
Yeah, thank you.
Okay, so I think with that we finish this last keynote, and well, we thank again Mark. And now?
They're driving fast cars, but they don't know where they're going. In the fast lane, living life without knowing. And he says, I wish that I could be like the cool kids, 'cause all the cool kids, they seem to fit in. I wish that I could be like the cool kids, like the cool kids. I wish that I could be like the cool kids, 'cause all the cool kids, they seem.
So this is the end. So we have a short ceremony to remind you of the few things we have done these days. Looks like years ago, but it's only a few days ago. And then we have the ceremony for the awards, the poster awards, and then the presentations for the future ISMB and ECCB conferences. So let's go. No, first of all, I have to say that we, as organizers, are extremely happy of having had you here for these days. We have, as you know, a lot of doubts about the chances of this conference to be successful with the many things that happened in the world in the last few months. And at the end, to have this room and the conference place full of young people, old friends and new friends has been really a very satisfactory experience, something that we are not going to forget. So I think that all the work that Salva, his team did to prepare that is by now, I would say, well paid in terms of satisfaction. And if you are happy, we are happy. So just a reminder, as you know, this is the 21st edition of the European Conference in Computational Biology. It's the second time this conference happened physically in Spain. If we count the 2 years ago virtual conference, will be the third time that we organize ECCB in Spain. So it's time to pass this to someone else, as we will do. We tried this, we are experimenting with ways of organizing conferences in this new time. In this case, we decided to do this hybrid mode. So we had, in the days before the presential conference, these days of virtual workshops that I think works very well. And we got a lot of people attending, a lot of people registered, and many of them attending the workshops from many different places in the world. We did this in the afternoon to have chances to have people attending the conference from America. And then we have these days of presential conferences. We didn't want to make this fully virtual, so we didn't want to have speakers on the screen only. So we were just broadcasting the talks, and as you have seen, there were a lot of people following them too, and we have questions from the people following the talk remotely, but then we have the pleasure to have physical speakers, three-dimensional speakers, what is makes things more interesting, and also the possibility for all of us to interact with the speakers. So numbers that we have by now, we almost got to the 900, so we missed 8 people. That's really a pity, say 900, but it's almost 900. With 181 virtual making, so the total number of people attending the conference is more than 1,000. That I think that is a good number. I think that in some point we need to meet with the other scientists, and I think we'll fulfill this well enough. 99 talks, so a lot of time to following talks, 500 posters, and a conference dinner. And I think we have a very good conference dinner. At least I like it. I hope you like it. I hear many people like it. I hear even people that came back after 4 in the morning. And I hope you also have time to enjoy Sitges. I think it's a nice city to visit. It's a nice city for tourists, but turned out to be, as far as I can say, a nice city for science too. So with this, I'm going to let Salva to introduce the highlights of the conference and the main talks. Salva.
Thank you very much, Alfonso. So we have the 5 traditional parallel tracks. They were very successful. So the room was almost all the time full. Sorry for that. We couldn't make it bigger. We have a very interesting new track on climate crisis and health. We expect that our colleagues for future editions keep it and expand it. So we have a very interesting debate, a panel debate with our colleagues from Latin America. So we're bringing the global community together, and I think it's important to work as a whole. The birds of a feather on sex and gender biases was quite interesting. It was a heated, I don't know if that is the right word, debate with more than 100 people participating. And finally, we have a number of posters. And that is really nice because it gives the opportunity to many of you to participate, to interact, to exchange knowledge. So before we close the conference, I have 2 or 3 announcements. First one, the people going to BSc The bus, the meeting point is in front of the technical secretariat. You will be guided to the bus. Please be on time. Second announcement, if you have any lost objects, in the technical secretariat, they have them. So a number of nice items have been found and deposited there. Please, let's be green. So if you plan to, please return your badge so we can recycle them rather than throwing them away. So we will do it properly. And do I miss anything? Okay, so my secret person here. So then, once again, we want to thank all the amazing keynotes, 6 keynotes during this conference. So the last one, Mar Albà, thank you, Mar, for the presentations and so on. We'd like to thank as well that the chair has made an amazing job during the conference, before the conference, and during the last 4 years. Many of them has been the chairs of the tracks in the last 2 editions, so it has been a great job. So thank you very much. The sponsors. Without sponsors, this conference hasn't been made possible or could have been a little bit more expensive. So thank you to our different sponsors. And I would like to take a moment to really acknowledge the work of many people. So Alfonso and I are standing here, but it's the work of really, really many people. In my team, I would like particularly to thank Laura. Laura, where are you, please? Can you come? Come over, please. So Laura has been the person dedicated for a year organizing ECCB, many emails, many hours, so please, come. Oh, sorry. And then I would like also to invite Eva. Come over, Eva. So thank you very much. Of course, I would like to thank Carmen. She is running away. So she's a representative from Visio. Thank you very much. And finally, I would like to thank the amazing group of volunteers that have been working or collaborating hard during the conference, guiding the chairs, and many of you across the different sessions. Thank you, guys. You know who you are. And now, before we finish, remember we can use F1000 to deposit your posters, your talk slides, and so on. So you get a DOI, your work can be credited, can be shared, and so on and so forth. And without further delay, we would like to announce the 3 awards for the best posters. So the competition has been hard. So we have a Eurovision system. So we have a panel, and then we have the popular votes. And I would like to invite to the stage Gonzalo Parra, please, to present the posters. And then Javier de las Rivas, as a Goblet representative. Goblet has kindly agreed to sponsor the poster winners. So So I have to say that the third place will get a €120 prize, which is not good. Sorry, it's not bad. 150 for the second one, and 180 for the first one. So thank you, Goblet. Thank you, Javier, for making this possible. So third prize goes to Simon Cirani. Hopefully, Simon is here. So the poster title is a linear optimization approach for normalization of SARS-CoV-2 vaccination data. So Thank you for your contribution. Wait for the picture, wait for the picture. Look here. Oops. Yeah. Should I look at it? There? Perfect. Thank you very much. Congratulations. So, the next one, the second prize goes to Joana Pereira for a poster titled, "What is Hidden in the Darkness? A large-scale approach to make sense of all natural unknown proteins." So, Joana, cover. So the prize is presented by Javier de las Rivas. Thank you very much. And finally, last but not least, the first prize goes to Brigitte Bowman for a key velo improved single-cell RNA velocity estimation. Brigitte. So we are really happy to see the 3 winners in the ceremony. Usually it doesn't happen, huh? So the prize is presented by Alfonso Valencia, the conference chair. Congratulations. Thank you very much. So the next ISMB ECCB meeting will be 2023 in Lyon. So we have prepared a video for all of you to grasp what is this edition about. So please, the video. And now is the time to announce ECCB 2024. So we go a little bit far north, but before that, please, Tommy, can you join us?
Hello, good afternoon. I'm Tommi Nyränen. I'm the head of node of Elixir Finland, and I'm really proud to pronounce and announce that Finland will be hosting the ECCB 2024. It will be hosted by CSC, my home organization, and University of Turku. My colleague Laura Elo, who is the chair of the scientific committee, couldn't be here, but sends her warm regards. I was thinking really hard what to say about Finland after this Spanish experience. The standard has been set really high, and the video from Lyon. So I think in Finland we can only promise at this point that it will be cooler. Anyway, the ECCB 24 tradition is that we commit to organize this for you, this outstanding conference in Finland. And we have also prepared a 1-minute video about the key facts that you should know at this point. Please.
Turku, the oldest city of Finland, the city which is known for its science and research.
Finland hosts Lumi supercomputer, one of the fastest and coolest and most environmentally friendly research instruments in the world.
With computational and multidisciplinary expertise, together we can respond to the growing global challenges.
In ECCB 2024, you can make your mark how this development goes forward.
Turku is easy to reach by train, plane, or sea. And while staying in Turku, consider the eco-friendly alternatives, city bikes and electric scooters, to move around in the city.
We look forward to welcoming you to Turku in September 2024.
No niin, laitoimme saunan ja olutta on. In other words, welcome to Turku and follow the website, so we'll update things there. Thank you.
Thank you. Thank you, Tommy. Tommy is becoming quite an actor for what we see. So, dear friends, that's the end. Thanks very much. We hope you go back home safe, happy, and with the head full of thoughts and things to do in the future. It has been a pleasure to have you here. Bye-bye.